Hello, my name is Alastair King and I'm the Head of Biology at Eurofins Labs. It's a great privilege to be able to present to you today aspects of Adme and how that is important in drug discovery and the various assays that can be used to address Adme. I'm very grateful to the organizers, the Biological Research Information Center, in order to be able to present this webinar. This is an overview of the agenda that we will cover today. We will start with taking a look at Adme, what is it and how is it important in drug discovery. We will then look at some of the assays that can be used to address the different aspects of Adme. This includes looking at compound solubility, binding of compounds to proteins, metabolic stability of compounds, absorption and permeability of compounds, and lipophilicity of compounds. These are all important properties of compounds that can affect the overall Adme picture and thus also overall affect the availability of a drug when being used as a therapeutic. We will then take a quick look at later stage Adme assays for later stage drug discovery efforts. We will look at the various Adme assays and how they can be used together in a screening cascade. We will take a quick look at a couple of case studies, and then we will end with a summary of what we have learned today. In terms of overall drug discovery, there are lots of different activities that must be addressed and pursued in order to develop a medicine that has the greatest efficacy, safety, bioavailability, and overall properties that are suitable for development into a drug. For small molecules, obviously, this starts with chemistry, so the synthesis of various molecules that can be then evaluated in a variety of assays and in an iterative procedure to gradually optimize those molecules until one has a candidate drug. Obviously, activity in vitro assays is important for this, and one has to be able to dial in the appropriate activity, both in terms of potency and selectivity, at the desired target or targets that are relevant for the particular disease being addressed. Tied in with that are aspects of safety pharmacology. A drug must not be capable of modulating other targets that are considered off targets or undesirable targets, where the activity at those targets could result in toxic or unsafe side effects. A drug may well work in an in vitro biochemical assay, but of course it also has to work in a cellular environment. So translational phenotypic assays are very important, not only in terms of identifying how a drug will work in a phenotypic setting, and that would be typically modeling the disease in an in vitro sense, where one is looking to see if the drug can work and modulate different aspects of biology that are relevant to reversing that disease state. But one also needs to get an idea from a cellular perspective of the consequences of that drug, and that may include aspects of safety. A drug obviously has to work in an in vivo system, and this is obviously a lot more complex than any in vitro biochemical or cellular assay. And because of the complexity of the living body, these are models that are used to address how a drug gets to where it needs to go in the body to do its job as a potential therapeutic. Obviously, any drug discovery program in some cases requires custom assays and various products, and these are available to assist the drug discoverer in the pursuit of all of these activities. Now, we will be talking today about Adme Tox, shown at the bottom of the slide here, and this is just as an important aspect of drug discovery as any one of the other categories that I've just mentioned. Adme Tox is the combination of the acronym Adme, and we'll talk about that in a second, as well as evaluating potential toxicities. Now, these properties are all part of the overall profile of a drug. In particular, the Adme side of the properties delineate how a drug gets to where it needs to go in the body, and so makes that vital link between in vitro observations and how a drug behaves in vivo, as well as how that drug may cross into the appropriate tissues to get to where it needs to go in order to have its therapeutic effect. It also addresses how the body tries to get rid of a drug by clearing it, typically in the urine, and that particular activity may result in metabolic products that could have either beneficial or toxic effects. And collectively, the whole process of Adme will look at what is called the bioavailability of a drug. So the different aspects of Adme all contribute to how stable a drug is, if it gets to where it needs to get to in the body, and if it lingers or it lasts for a long enough time in order to be able to do its job as a therapeutic. So what is Adme? Adme, as I'd mentioned, is an acronym which stands for absorption, distribution, metabolism, and excretion. So for any drug to be able to do its job in the body, it has to be absorbed, and that may be absorbed from oral dosing, or it may be absorbed into appropriate tissues. And in order to do that, the drug has to cross various barriers. And some of the examples I have listed here include the intestinal, nasal linings, lungs, or skin. For example, an inhaled drug has to be able to cross the barrier from the lungs and get into the body that way. A topical agent must be able to cross through the skin to get into the body to do its job. Distribution is important because the distribution actually outlines how well the drug can be distributed to other parts of the body. For example, a drug may be taken as an oral drug, but it might need to get to the brain in order to do its job. So that drug then needs to be able to distribute appropriately to get to the tissues where the target for which it is designed resides and where the disease is to be treated. Metabolism, as I'd mentioned on the previous slide, is an important aspect of Adme because metabolism outlines how a compound is broken down, typically by the liver. And this is done because the body sees a chemical agent as a foreign entity, and it will do everything it can to try and metabolize that foreign compound so that it can be excreted and passed out of the body. Obviously, if a drug is metabolized very quickly, it may not be around long enough in the body to be able to do its therapeutic job. Alternatively, a drug may be metabolized very slowly, and that may be beneficial such that the drug does exist for a long enough time to be able to do its therapeutic job. But of course, also in certain other cases, if a drug exists for too long, it may result in toxic side effects. And that is one of the factors of drug-drug interactions that have to be considered. It is also possible that a drug may be metabolized into toxic metabolites. And this also needs to be addressed because the drug itself, when applied, may itself not be toxic, but if it is metabolized into toxic byproducts, this can cause a problem. A drug ultimately then is excreted, so collectively the Adme process describes how a drug enters the body, gets to the tissues in which it needs to act, is metabolized, and ultimately is excreted from the body. And the window in which that drug is present in the body is important in determining how well that drug can act. That window being both temporal as well as in a distribution sense. In terms of early drug discovery efforts, there are various Adme assays and approaches that need to be considered when optimizing drugs and developing them to carry forward in a drug discovery program. Solubility is very important because a drug needs to be soluble. It needs to be soluble because in that way then it can be administered and get to where it needs to go. If it is not soluble, this can cause problems in actually getting the drug to where it needs to go from an availability perspective. Tied in with that is permeability. So a drug needs to be able to cross not only cell membranes, but certain tissue types. I'd mentioned earlier the potential for a drug to be used to treat maladies in the brain. So that drug needs to be able to cross the blood-brain barrier in order to do that. But at the very least, a drug whose target resides within a cell needs to be able to cross the cell membrane and get into the cell to do its job. Drugs often bind to other environments in the body, and that can include fatty tissues, and also potentially when crossing a cell membrane, if a drug has a particularly high affinity for lipids, it can then cause a problem with the drug not crossing the membrane. And so Lipofelicity is another aspect of the Adme properties of a drug that needs to be addressed. Somewhat related to that, but from a different perspective, is protein binding. So a lot of drugs will bind to other proteins, typically plasma or serum proteins, and this can have a variety of different effects on the half-life or the existence of a drug within the body. If a drug binds to a protein, that may sequester the drug away from being able to do its job in a therapeutic sense. If a drug binds to a protein, it may also be that the drug could depot there, and if the drug is slowly released from the protein, that could also affect its half-life. So it may well be that a certain amount of protein will keep a drug around in the body for long enough. So protein binding is very much a balance in terms of where the drug is supposed to get to in the body and how long it lasts in the body to be able to do its job. And then lastly, as I'd mentioned on the previous slide, metabolic stability. The body will do everything it can to try and remove the drug. So if a drug is unstable in a metabolic sense, it will be metabolized very quickly and excreted, and thus may not be around for long enough to do its job in the body. Conversely, if a drug is not metabolized quickly, it will have typically a longer half-life in the body, and this may be beneficial, or it may not, depending upon what the drug is intended to do and for how long it really needs to be in the body to do its job. So collectively, all of these properties need to be addressed when evaluating the Adme properties of a drug, and optimal aspects of all of these properties contribute to what might be considered drug-like properties and are a very important part of the equation in drug discovery. So let's start by looking at compound solubility. Solubility is a very important aspect of the Adme profile of a drug, because if a drug is not soluble, it very likely won't get to where it needs to be to do its therapeutic job. So a compound with a lower solubility generally has a higher risk for failure for the various properties and the various aspects outlined at the top in italics. In some ways, a drug that is less soluble may give misleading data in vitro assays, either by indicating suboptimal potency in assays looking at targeted activity, or potentially by masking undesirable off-target activities, where, for example, a safety pharmacology type assay might be showing a lower activity that seems to be appealing, but that's actually a result of the poorer solubility of the drug. And obviously solubility can influence things like pharmacokinetic or PK properties, as well as pharmacodynamic or PD properties of the compound. And overall, this affects the developability of the compound. So compound solubility can have a direct impact on the Adme properties of a drug, and generally insoluble compounds have a number of issues that are related to that low solubility. Obviously, lower solubility can result in lower bioavailability. So if a drug is not as soluble, it may not get to where it needs to go in sufficient quantities because it simply isn't soluble enough. It may not be able to get to the right concentrations or to be administered at the right concentrations that are needed for the drug to have its effect. The drug may also have enhanced binding to plasma proteins. Under those circumstances, if the drug is partitioned more highly to plasma proteins as compared to the soluble matrix, then again there may be less drug available to reach its target, to get into the cells to do its job. And the other issue with low solubility is that a drug may have the tendency to precipitate out of solution, not only in vitro assays, as we'd mentioned a few seconds ago, that can give misleading results on both potency in desirable assays, as well as potential liabilities in the safety pharmacology perspective, but also if a drug has the tendency to precipitate out of solution, this may possibly occur in the body that could result in localized toxicities at the point at which it precipitates. So generally, insoluble compounds may need other formulation strategies if their other properties are desirable enough to want to take that drug forward. This can, of course, lead to a variety of additional costs when examining some of those other formulation strategies. And it can also, because of the time needed to evaluate these strategies, result in the delay in being able to advance that drug. So generally, it is advisable to evaluate solubility aspects of a drug early. And when one is looking at assays to address potency at the desired target, selectivity, and safety pharmacology, it is wise to look at various Adme properties, including solubility, in parallel with that, such that as medicinal chemistry advances are made with the development of a compound, not only can the properties relating to its fundamental biological activity be improved, but so can its Adme properties, and that includes solubility. So there are a variety of factors that affect the solubility of a drug. This includes things like molecular weight. Generally, the larger a drug is, the less soluble it will be. And this is one of the aspects of the Lipinski rule of 5 that medicinal chemists will employ when optimizing a drug for better Adme properties. Lipofelicity can also affect the solubility of a drug. If the drug partitions preferentially into a lipid environment, then less of that drug will be available in an aqueous environment, as represented by the cell or the surroundings around a cell, and thus there will be less drug around to do its job. Particle size can also affect solubility, and generally the larger the particle size of a drug, the less soluble that particular drug may be. And then different salt forms, different solid and salt forms of the compound can also affect its solubility. For example, a drug, either as something like a free base or a hydrochloride salt, may have relatively poor solubility, but upon conducting a salt screen, it can be found that there are other salt forms of the drug that have improved solubility. And a good example of that might be the case where a drug as a hydrochloride has poor solubility, but then when formulated as a tosylate or a mesylate salt, has much greater solubility. And then last but not least, the acid dissociation constant, or pKa, is another important factor that can affect solubility of a drug. Obviously, a drug, depending upon its route of administration, may be exposed to different extremes of pH. For example, the very low pH in the stomach, the mild alkaline pH in the lower intestines. So this can also tie in with the solubility of the drug and thus its availability. So there are different ways, different assay formats that can be used to evaluate the solubility of a drug. And this includes looking at kinetic solubility as well as thermodynamic solubility. So there are different ways of looking at compound solubility, and Eurofins provides a variety of services that allow this to be addressed. Solubility can be different in different environments or different matrices within the body. And one of the ways of doing that, and assessing solubility in those different matrices, is to simulate those environments. So for example, simulation of the gastric fluid with a very low pH can allow one to address the solubility of a drug when it enters the stomach. Simulation of the intestinal fluid is another way of looking at solubility when that drug reaches the lower intestines. And we can actually offer a number of services to look at client-specific options. So, for example, solubility of a drug in particular matrices that relate either to the route of administration of a drug or to the specific tissue type in which the drug is ultimately to be effective. There are a variety of different ways of looking at solubility, and these include things such as HPLC or high pressure liquid chromatography coupled with a UV trace or laser nephelometry. So collectively, we can look at the solubility of a drug in different environments and via a number of different methodologies to provide those necessary readouts. So we'll now take a look at protein binding. Drugs can bind to a variety of proteins in the body, and that binding can have a great effect on the amount of free drug that is available and its pharmacokinetics. Drugs can bind to a variety of different proteins, and these include things such as serum albumin, alpha-1 acidic glycoprotein, a variety of different lipoproteins, and several other different biological moieties, and that includes things such as red blood cells. So when one thinks about protein binding, a drug can exist in one of two forms, either as the free drug, where it is able to then bind to its therapeutic target and exert its therapeutic effect, or as a complex with the protein to which it binds, where it typically is not able to exert its effect at its target. So a drug has affinity for different proteins, and the degree to which that drug binds to a protein dictates the amount of free drug that is available to then bind to the target. So the more strongly a drug binds to a protein, the less amount of drug there is available. So effectively, the free drug concentration is lower. And this really happens because drug binding to proteins exists as an equilibrium. So this is something that really has to be considered as part of the Adme profile of a drug. And protein binding, in effect, can be a very significant factor in determining the biological half-life of a drug for a variety of reasons, not least of which is if a drug binds to a protein, it will stick around longer in the body and it is less likely to be cleared. But of course, this remains a balance then between extending a drug's half-life by binding to a protein, but it's also being able to act at its therapeutic target while not being cleared out of the body. Eurofins offers a variety of services to evaluate protein binding, and this is shown here, examples being looking at protein binding in not only human, but also a variety of different animal species. And this can be useful when examining animal models for PK. This is obviously a necessary part of drug discovery, and so evaluating the ability of a drug to bind to different proteins in different species can help rationalize the toxicities and the efficacy profiles that are delineated in models using those different animal species. So one can use equilibrium dialysis in a microplate format, and that can be used to determine the amount of bound versus unbound drug when exposed to these different proteins. So we can do this using a variety of different proteins, including those that are present in plasma, in serum, or particularly albumin, or a variety of other different client-specified matrices, as we'd mentioned earlier. So let us now look at metabolic stability. This is a measure of the degree to which a drug is metabolized, frequently by the liver, and this can have a profound effect on the drug's Adme profile. If a drug is metabolized very quickly, it is cleared quickly, so the drug may not exist in the body or in the target tissues long enough to exert its therapeutic effect. Alternatively, if a drug is metabolized very slowly, while it may exist longer to exert its therapeutic effect, this may also result in a variety of toxicities due to that prolonged exposure. And in fact, there are many drugs that have never become good approved drugs due to a poor metabolic profile. So understanding how a drug can be metabolized, and this is sometimes referred to as biotransformation, is an essential part of the Adme evaluation of a drug, and thus the process by which drugs are selected to become candidates based on favorable pharmacokinetic profiles. So metabolic stability assays that are in place can effectively measure what is called the intrinsic clearance of a compound. And that is the way in which that compound is metabolized, as I said, typically by the liver. And it provides key data that can then determine the pharmacokinetic parameters of the drug. And this includes such parameters as bioavailability and half-life. For example, if a drug is metabolized very quickly, the half-life will be commensurately lower, whereas if a drug is metabolized slowly, its half-life will be longer. As I'd mentioned, the liver is probably the most important site for drug metabolism in the body. And in fact, the cytochrome P450 enzymes, or the CYP enzymes, are key to being responsible for that metabolism. So there are a variety of assays available that can address and model different aspects and ways in which the liver metabolizes various drugs and gets rid of them out of the body. This includes looking at things such as liver microsomes, hepatocytes or liver cells, S9 fractions, as well as aspects of blood and plasma, and in fact even recombinant enzymes, for example recombinant cytochrome P450 enzymes. And examples from not only human sources, but also from animal species allow one to collectively demonstrate the metabolic profile of a drug in both the human system, obviously from the therapeutic perspective, but also in the animal system that allows one to rationalize PK and PD responses as well as toxicity responses in those various animal models in which the drugs are being evaluated. Eurofins offers a number of different metabolic stability assays, so these measure the CL-int, or the intrinsic clearance value of a compound. And these data can be used to generate a variety of parameters, and this includes, as I'd mentioned previously, the bioavailability and the half-life. So we actually have a number of assays available that model the clearance based on those different in vitro systems. That includes liver microsomes, hepatocytes, S9 fractions, blood and plasma, and recombinant enzymes. And this uses both sources from human as well as animal species. So performing intrinsic clearance assays provides another component of the Adme profile of a drug that collectively outlines overall its bioavailability. Another aspect of Adme testing is absorption and permeability. A drug obviously has to be absorbed from its route of administration, whether that's through the lungs in a nasally inhaled drug, whether it is through the GI tract following oral administration. And a drug ultimately has to cross membranes in order to get into a cell where it conducts its activity as a therapeutic, or to cross into specific tissue types. We'd mentioned earlier about the ability of a drug to cross the blood-brain barrier if it is designed to modulate a target in the brain. So oral administration is frequently a preferred route for the majority of drugs that are developed. Taking a pill is a lot easier and gives a greater quality of life than having to administer a drug using IV or intravenous administration, and especially in cases like chemotherapeutics where a drug may need to be given frequently. So there are different aspects then of absorption and permeability that can be modeled. And in fact, those models can evaluate different aspects of how a drug gets into a cell or crosses certain barriers. So we have assays that look at things like artificial membranes. So the parallel artificial membrane permeability assay, frequently abbreviated as PAMPA, is one such way of doing this. We also have assays to look at the ability of a drug to cross a cell monolayer, and frequently use models that are industry standards, use CACO2 cells or Madin-Darby canine kidney cells, MDCK cells. So the permeability of a drug depends on a number of different things. This includes things like lipophilicity, or the log P. And so if a drug is highly lipophilic, it has the potential to get into and cross the membrane. But of course, if the drug is too lipophilic, it may not actually get through the membrane. Permeability also depends on the acidic dissociation constant, or the basic dissociation constant, the pKa and the pKb values. And so effectively, only an unionized drug will cross the membrane. If a drug is highly charged and remains that way, it is not going to be able to cross through the lipid part of the membrane between the two surfaces. Intestinal absorption of a compound is also dependent upon a variety of things. That includes permeability, so the ability of the drug to get out of the GI tract, across the GI barrier, and into the rest of the body. It depends, as we discussed earlier, upon solubility and also dissolution. So there are various ways in which a drug can be modified as part of the optimization of a drug through medicinal chemistry to improve its properties and thus improve its permeability. This includes things like reducing the ionizability of a drug, adding in a little bit of lipophilicity so that the drug is able to freely cross membranes and get to where it needs to go, reducing the polarity of a drug, and reducing its size. The larger a drug is, the less likely it is to be permeable. And again, this comes back to the Lipinski rule of 5 that we mentioned earlier. And also the potential of constructing a drug as a prodrug. This also ties in to some degree with metabolism. There are a variety of drugs that are administered as a prodrug that in of themselves are not active, but this can sometimes result in properties that allow things like permeability and allow a drug to get to its target, where it is then processed or metabolized into an active form. And a good example of efforts in this area might be a drug that is a prodrug that is needed at a higher local concentration, for example in a tumor. And so that prodrug is designed such that when it gets into the tumor, it is processed there, providing concentrations of the drug that are suitable to provide anti-tumor efficacy. Other examples of Adme testing that we can do to address things like absorption and permeability are shown in the various assays that Eurofins offers. This includes the industry standard models with human CACO2 cells, as well as the MDCK canine kidney cells. We also have the PAMPA assays available there as well. So collectively, we can provide a complete portfolio of assays to evaluate how a drug is capable of being absorbed and its permeability from a cellular perspective. The next topic that we should explore is lipophilicity. This is a key determinant of the pharmacokinetic or PK behavior of drugs, and it can affect a wide variety of Adme profiles, including distribution into the tissues, absorption of the drugs, as we discussed on the previous few slides, and the binding characteristics of a drug. And it's also a key determinant in the solubility of a compound. As we discussed earlier, if a drug is highly lipophilic, it is less likely to be soluble in an aqueous environment. If a drug is highly lipophilic, it may also be very easy to get it into the membrane of a cell, but not necessarily through it. So, in a balance, a compound with a moderate lipophilicity has a good balance between its solubility and its permeability, such that it is soluble enough that it can exist as a free drug in circulation or in the aqueous medium inside a cell, but it also has enough lipophilicity that allows it to get into the tissues that it needs to get into to exert its effect, as well as to cross the cell membrane into cells. So this is a key part of the Adme profile for a drug. And a good example we've mentioned a few times in this presentation is the ability of a drug to get across the blood-brain barrier where that is needed. Obviously, lipophilicity also can affect the metabolic stability of a drug or its liability for high metabolism. So thinking about it in terms of the two extremes, if a drug's lipophilicity is too low, this can result obviously in high solubility, but at the price of very low permeability and metabolism. If a drug's lipophilicity is very high at the other end of the spectrum, this obviously results in a much lower aqueous solubility, but potentially then higher permeability, but also metabolism associated with that. A way of measuring lipophilicity is called the log D, otherwise known as the distribution coefficient. This is a measure of how lipophilic a drug is. This is typically looked at by seeing the distribution or the partitioning of a drug between an organic solvent, typically octanol, and an aqueous buffer. So the more lipophilic a drug is, the more it partitions into that organic solvent, and the less drug there is in the aqueous buffer. Conversely, the less lipophilic a drug is, the more the drug will appear in the buffer, the aqueous buffer, and not the octanol phase. So this is what is used to determine the lipophilicity or the log D value of a drug. Now, it's not the only method. There are other methods that are available for the evaluation of lipophilicity, and that includes things such as HPLC-UV. Eurofins offers a variety of assays that are designed to look at the lipophilicity of a drug. And an example here is shown with the log D of a variety of different drugs by partitioning into octanol versus the aqueous phase. So again, this uses a microplate format with a shake flask technique. So ensuring good opportunity for the drug to partition into either the octanol or the aqueous phase. And then measuring the amount of drug in each of those phases then allows calculation of the log D values. So in doing that, HPLC-UV analysis is one of the ways in which the compound's lipophilicity is determined. So we've now covered the various aspects of Adme testing that are typically used in early Adme testing, and really all of those parameters should be evaluated frequently and from an early stage so that one can begin to bring the best Adme properties and qualities into a molecule as one goes through drug discovery. But one then faces a situation where one goes to a more in-depth evaluation, and this is typically conducted in order to satisfy a variety of regulatory requirements. So the various regulatory agencies require looking at Adme properties in a lot more detail. And this effectively allows a much fuller and broader understanding of the compounds from their Adme property perspective, and in many ways is looking at different aspects of some of the basic Adme properties that we've seen previously in much greater detail to understand how that Adme profile is manifested. So this is particularly important when preparing for preclinical candidate selection, for preclinical development, and ultimately for going into clinical studies, which is why the regulatory agencies are very interested in looking at the Adme properties of a drug in greater detail. So this encompasses things like looking at P450, the cytochrome P450 enzymes in greater detail. We discussed those earlier as being key drivers of metabolism of a drug, but there are various aspects of Adme that can relate to P450 biology. One can look at the different kinds of cytochrome P450 enzymes that metabolize a drug, but a drug can also itself have an effect on the P450 enzymes itself. A drug can, for example, inhibit a P450 enzyme. And this can be significant if the patient ultimately is taking other drugs that themselves may be metabolized by that P450 enzyme that is inhibited. Drugs can also induce the expression of P450 enzymes. So combined between inhibition and induction, these are ways in which a drug can actually modulate the ability of the liver to process perhaps itself, but also other drugs. And so that can potentially upset the imbalance of the system if a patient is taking more than one drug. There is also the ability to assess transporters, so ways in which drugs can get into and out of cells. And there are a variety of assays that are available to evaluate transporters and which transporters will typically carry a drug across the cell membrane. There is also metabolite profiling. So metabolite profiling looks at the ability of the liver system typically to metabolize a drug into what might be potentially toxic metabolites. And this is important in understanding what the liabilities of a drug may be. So we can profile the effect of drugs in a variety of different ways with respect to their ability to inhibit or to be substrates of the cytochrome P450 enzymes. Cytochrome P450 enzymes represent the phase 1 form of metabolism, whereas phase 2 metabolism is mediated by enzymes such as glutathione S-transferase or UDP glucuronoside transferase. And collectively, these enzymes are resident in the liver, and they constitute the major mechanisms of drug metabolism throughout the body. Now, a drug can interact with cytochrome P450 enzymes in a variety of different ways. A drug may, for example, be metabolized by a cytochrome P450 enzyme, and it's important to understand the profile of metabolism. For example, knowing which cytochrome P450 enzymes can metabolize that drug. This kind of evaluation is called cytochrome P450 or CYP phenotyping. Alternatively, a drug could be capable of inhibiting the activity of a cytochrome P450 enzyme. So this is known as CYP inhibition. And this is particularly important for patients who may be taking multiple drugs. So if the drug candidate being evaluated is capable of inhibiting a particular cytochrome P450 enzyme, then that may result in aberrant exposure of the other drug that a patient is taking. And it may result potentially in aberrantly high levels of that drug, which could lead to toxicity or safety issues. Drugs can also interact with a variety of nuclear receptors, which themselves are transcription factors, and ultimately can modulate the expression of different cytochrome P450 enzymes. So the ability of drugs to modulate cytochrome P450 expression is what's called cytochrome P450 or CYP induction. So Eurofins offers a variety of different assays to evaluate CYP inhibition and CYP phenotyping. There are a number of different assays available to look at the variety of different CYP enzymes, including assays for 3A4, 2B6, 2D6, 1A2, 2C8, 2C9, and 2C19, as well as a variety of other different isoforms. And these assays use a variety of different drug substrates, either as a reference or in the case of inhibition studies, to see whether a drug is capable of inhibiting the activity of those enzymes. And that's actually shown here on the right-hand side, where there is a profile for CYP3A4 looking at ketoconazole in the presence of different drugs or testosterone. So collectively, we can evaluate then the effect of drugs either as substrates for CYP450 enzymes or in their own ability to modulate either the activity of a CYP450 enzyme or to modulate that CYP450 enzyme's expression. So this is a key part of understanding the Adme profile of drugs at the tier 2 level. So looking a little more closely now at CYP induction. This is the process by which a drug is potentially capable of modulating the expression of a CYP enzyme at the level of the nuclear receptors that are capable of regulating the expression of the gene for that particular CYP enzyme. So if a drug is capable of affecting a nuclear receptor, which is effectively a transcription factor, that can result in changes in the level of a particular CYP enzyme. And that's actually summarized in the graph on the right-hand side here, where one sees a time-dependent induction of CYP expression at the level of mRNA that is caused by the various drugs that are shown. So for example, looking at rifampin here, one can see that over the time course from 6 hours to 72 hours, there is a dramatic increase in the expression of the CYP3A4 gene in this particular case. So CYP induction assays are something that Eurofins offers, and that allows one to evaluate the effect of a drug on modulating CYP levels. And this is done using qPCR methodologies to look at the transcription of genes relevant to those different cytochrome P450 enzymes. We can also look at transporter assessments. So a drug's ability to get into a cell or to be shunted back out of a cell is a consequence of its ability to act as a substrate for a variety of transporters. Now, again, in the same way that we looked at CYP activity, both from the perspective of being a substrate, but also inhibiting those CYP enzymes, we can do the same thing with transporter assays. So a drug may be a substrate for a particular transporter, and that can obviously affect the uptake or the efflux of that drug into or out of a cell respectively. But it can also result in toxicity by modulating the effect of those transporters that could affect other drugs as well. So Eurofins offers a variety of transporter assays across a fairly broad portfolio of different transporters. And this can evaluate whether or not a drug interacts with a key transporter, either from the perspective of being a substrate or an inhibitor. And so we can look at different transporters in different classes. For example, there are the ABC type transporters, as well as the solute carrier or SLC transporters. And we can do this in a high throughput format. So this allows rapid profiling of a large number of compounds. And we can do this in cellular backgrounds, for example, using CACO2 cells, MDCK cells, HEC293, and CHO cells, as well as a variety of artificial membrane vesicles and a variety of transfected cell lines. The last part that we will look at here is looking at metabolite ID and profiling. So as we discussed earlier, drugs can be metabolized as part of the body's attempt to clear them from circulation. And in doing so, the drug can be metabolized into a variety of products. Sometimes the products have no consequence and are rapidly cleared from the body, but sometimes a drug may be metabolized into something that has a toxic effect. So when one looks at metabolism, one can look then at the spectrum of products that are generated. And this can be done in a clinically relevant sense, looking at clinically relevant metabolites that are not necessarily evaluated in non-clinical studies. In certain cases, toxic metabolites might be formed in both the human or in animal species. And understanding metabolite identification and the profiling of those metabolites can go a long way in certain cases to rationalizing toxic effects that might be seen in an in vivo animal model. So one of the things that Eurofins is capable of offering is a full metabolite profiling and structural identification in a variety of different matrices. As we discussed earlier, this can include different environments, including things such as liver microsomes, liver S9 fractions with cofactors added, looking at hepatocytes as examples of isolated liver cells, or looking in plasma. And we can do this in both the human as well as a variety of different animal species situations. What is key to this are extensive mass spectrometry capabilities, and they really drive the ability to separate out and to evaluate the different metabolites that are formed. We will now take a quick look at how Adme assays are used in a typical screening cascade. Like any drug discovery assay, no one Adme assay should be used in isolation. And in fact, as we've discussed over the previous slides, there are many different aspects of an Adme profile which need to be addressed in the optimization of compounds to become suitable drugs. For all targets, there are various things that do need to be assessed, such as solubility, hepatic microsomal stability, and also how the drugs interact with cytochrome P450 enzymes, as we've just discussed on the previous slides, as well as their ability to be metabolized. But there are certain cases where particular assays in the Adme portfolio are more relevant. A good example would be for drugs that are orally administered, where one is looking at local and gastrointestinal features of a drug's stability, or its metabolism, or its solubility. These include things like the PAMPA assay, the simulated gastric fluid or simulated intestinal fluid solubility and stability studies, as well as gut S9 stability. When looking at something like central nervous system applications, the MDCK-MDR1 transporter assay would be a suitable assay to include in a screening cascade. Also, things like plasma protein binding and brain homogenate binding would be more relevant there. And collectively, the appropriate assays to be used in different environments, along with those which are more generic and applicable to all environments, collectively can help rationalize and understand the observations seen in vivo models, whether this relates to efficacy in an in vivo model or safety and toxicity aspects in an in vivo model, and relating those back to the amount of free drug that is available and that could be causing either those positive efficacy effects or those negative safety and toxicity effects. So looking briefly now at toxicity, this is an important part of the overall Adme tox or Adme and toxicity evaluations that one can and should do for a part of a drug discovery program. Toxicity can manifest in a variety of different ways. This can include toxicity in the liver, toxicity in the heart or cardiac safety toxicity, renal toxicity, and also genotoxicity. So these represent different areas that are key in the ability to de-risk a drug candidate for its potential to cause adverse effects later on in an in vivo system or ultimately in a clinical trial. So there are models that can address all of these different toxicities, and these are important to include in a drug discovery campaign. These models are in vitro models, and they encompass a variety of different cell types. And these cell types are relevant to the toxicities that are outlined. There are also a variety of other safety pharmacology panels available, and these constitute either in vitro biochemical or cellular safety assays that can be used to look at the liability of a compound with respect to targets that are known to have implications in a wide variety of different safety issues. So shown here are a number of the different safety panels that are available across Eurofins in our discovery portfolio. All of these assays should be considered as part of the development and discovery and identification of better medicines for the treatment of diseases. So we'll now take a look at where these Adme assays fit within the drug discovery process. This is shown at the top of the slide, going from target selection through lead finding or screening, characterizing hits, and then allowing lead prioritization, identifying suitable lead series for taking forward into the process of lead optimization that ultimately culminates in the identification of a candidate molecule to take forward into development and clinical evaluation. As we discussed earlier in this webinar, the Tier 1 Adme assays are well suited to the earlier part of drug discovery. They're high throughput assays, and they allow the rapid and facile evaluation of a large number of compounds to give a broad general evaluation of the Adme properties of those compounds. This helps enable lead prioritization and lead series identification. And this is really where Tier 2 Adme assays begin to take over. Those assays provide a more detailed evaluation of the Adme profile of compounds, which is especially important as one goes through the process of lead optimization, and ultimately because of their requirement by the regulatory agencies as part of an IND or investigational new drug application, are very important in being able to provide that package of information. Tied closely with this, of course, are the in vitro tox assays, These are best used in the area shown that starts in lead finding and going through into lead optimization. And collectively then with the in vitro safety pharmacology assays, the Adme assays provide a more thorough understanding at the level of the IND application of how a drug is exposed in the body, how it is metabolized, how long it lasts in the body to be able to conduct its therapeutic activity, what toxicity profile there may be, and what the safety pharmacology profile of that drug looks like. These properties also help rationalize preclinical animal models and to explain potentially how safety observations may be related to the exposure of a drug in those models. In summary, we've learned how we can profile the Adme properties of compounds in the pursuit of new drugs. That is to look at their absorption, distribution, metabolism, and excretion profiles, as well as their toxicology profiles. There are a great many assays available to look at these properties from an in vitro perspective, and Eurofins offers a comprehensive portfolio of these assays to enable the drug discoverer to develop better, safer, and more efficacious medicines. We back this up with a tremendous amount of experience in terms of consulting, designing, and conducting these various assays, and we've served a wide variety of clients from pharmaceutical to academic institutions. We have scientists with great expertise in the areas of DMPK and toxicology, coming from industry backgrounds, and we can provide cost-effective, high-quality, and reproducible data with fast turnaround times. This enables more efficient drug discovery processes and helps in getting better medicines into clinical development and ultimately into the clinic to be able to treat patients in need. We offer flexible, customized solutions to fit a wide variety of specific testing needs, and all of our Adme and toxicology assays can be modified to suit specific project testing needs. So in consultation with our scientists, we can then work with you to develop those specific assays that address those needs. So combined with our industry-leading safety pharmacology, in vitro pharmacology, ion channels, kinases, and in vivo models, and a variety of chemistry services, which we can also offer collectively as a start-to-finish drug discovery program that is managed under our Discovery One campaign offerings, we can provide end-to-end drug discovery solutions that employ all of these aspects. So with that, I'd like to thank you very much for your attention, and it's been a privilege to be able to discuss various aspects of Adme with you. Thank you.